Alzheimer's disease is defined by amyloid plaques, but amyloid does not physically dismantle a synapse, nor does it weave a tau filament. Stopping the clinical syndrome requires us to identify the specific enzymatic effectors that carry out these physical failures. Both events, the structural collapse of the dendritic spine and the mislocalization of tau, occur within a single anatomical boundary, the soma dendritic compartment. Over the last 15 years, two mechanistic proposals emerged from a single laboratory to explain how this compartment fails. Solving Alzheimer's requires a precise mechanistic account of execution to complement our understanding of onset, making these two dendritic proposals the center of our analysis. The first proposal, the synaptic brake model, suggests that a developmental repressor of excitatory synapses, called Ephexin5, is pathologically re-expressed in the adult brain. The second, the Sieve model, identifies a specialized membrane-bound proteasome that holds newly synthesized tau below its polymerization threshold. A decade of testing, spanning single-spine biosensors to human Mendelian genetics, has forced a complete revision of both models. These revisions ran in opposite directions, weakening the synaptic model's therapeutic logic while elevating the proteostatic model to a primary disease initiator. The two models merge into a single statement about a catastrophic failure of protein degradation in the Alzheimer dendrite. The initial premise of the Ephexin5 pathway is a matter of developmental restraint. In the immature brain, the Rho-A exchange factor Ephexin5 actively prevents excitatory synapses from forming. To allow spine growth, FB2 receptor signaling phosphorylates Ephexin5, licensing its destruction by the ubiquitin ligase UB3A. The disease claim held that amyloid beta oligomers strip the FB2 receptor. This stabilizes Ephexin5, which then accumulates and collapses spines through hyperactive Rho-A signaling. This logic appeared sound in vivo. Reducing Ephexin5 in amyloid overexpressing mice successfully rescued both dendritic spine loss and behavioral deficits. This linear cascade supported a clear therapeutic prescription: systemically reduce Ephexin5 levels to protect Alzheimer's synapses. Single-spine live imaging reported in 2025 revealed that Ephexin5 does not function as a simple Rho-A selective brake. Ephexin5 functions as a switch, activating Rho-A to drive collapse while simultaneously driving CDC42 to sustain activity-dependent spine growth. The choice between these two substrates is dictated by the phosphorylation state of a single tyrosine residue, Y361. When Y361 is unphosphorylated, the gate switches to the green, growth-promoting CDC42 output. The Y361 residue is the site that licenses UB3A degradation, while simultaneously activating the red Rho-A pathway. This dual function means that Ephexin5 knockout neurons fail to sustain growth during long-term potentiation because they lack the CDC42 arm. Ephexin5 abundance and its structural activity are mathematically locked variables. Systemically removing the protein ablates the neuron's own mechanism for activity-driven structural repair. In 2023, the evidence shifted from mouse physiology to human genetics with the discovery of Mendelian mutations in ARHGEF15, the gene encoding Ephexin5. Heterozygous loss-of-function mutations in this gene co-segregate completely with an autosomal dominant hereditary cerebral small vessel disease. This disease is caused by a chronic Rho-A and ROCK2 inactivation within vascular smooth muscle and endothelial cells. Suppressing this exact Rho-A axis is the proposed therapeutic intervention for neurons in Alzheimer's. The proposed strategy to lower Ephexin5 is an exact mechanistic phenocopy of a dementing human cerebrovascular disease. The second proposal centers on the neuronal membrane proteasome, or neuroproteasome, which manages rapid protein turnover at the synapse. This complex differs from the canonical 26S proteasome. It operates without 19S regulatory caps, without ATP, and without recognizing ubiquitin tags. Its substrate specificity is restricted to ribosome-associated nascent polypeptides, proteins caught in the act of being synthesized during neuronal stimulation. It functions as a homeostatic sieve, scaling the destruction of newly made proteins to match the rate of activity-induced synthesis. The neuroproteasome provides a specialized ubiquitin-independent disposal route localized specifically to the dendritic plasma membrane. New findings published in 2026 upgrade the neuroproteasome to a direct initiator of tau pathology. A selective inhibition of the neuroproteasome directly triggers the de novo formation of endogenous, sarcosyl-insoluble paired helical filaments. These filaments form in vivo in just 3 days, requiring strictly new protein synthesis with zero amyloid or seeds present. The surface abundance of this complex is modulated by apolipoprotein E isoform and declines with age. The quantitative thresholds scale with risk. ApoE4 neurons form filaments at 20% surface inhibition, ApoE3 at 60%, and ApoE2 neurons resist until 85%. This provides a comprehensive mechanism for tau initiation that scales with the greatest genetic risk factor for the disease. The Ephexin5 and Neuroproteasome models appear to describe two outputs of the exact same compartmental failure. Single-molecule localization clearly establishes that tau is translated almost exclusively in the dendrite, the same compartment where Ephexin5 is regulated. From 2017 data, we know neural activity drives a rapid proteasome-dependent reduction of Ephexin5 in dendrites. The neuroproteasome is the activity-dependent dendritic proteasome, dedicated to that rapid clearance. This suggests a unified hypothesis. The surface neuroproteasome clears both newly synthesized tau and activity-regulated Ephexin5. In ApoE4 genotypes, reduced surface proteasomes cause Ephexin5 to accumulate after activity. Tau polymerizes into filaments, while Ephexin5 locks into the destructive Rho A state. ApoE4-mediated neuroproteasome loss provides the missing mechanism for why the principal genetic risk factor makes spines fragile, unifying tangles and synaptic collapse into one lesion. This unified model requires a complete inversion of standard pharmacological approaches to Alzheimer's. Targeting the spine through systemic ROCK inhibitors or Ephexin5 degraders will inevitably induce cerebrovascular liability and completely block synaptic repair. Even existing proteostatic therapies pose a risk. Systemic 20S proteasome inhibitors used in oncology are mechanistically tau-initiating agents in neurons. Pharmacology must focus on upregulating, not inhibiting, enzymatic activity at the membrane. Viable handles include enhancing the trafficking of the complex, stabilizing its membrane anchor, or shifting ApoE4 behavior toward the ApoE3 state. Preventing the structural execution of Alzheimer's depends on recognizing the dendrite as a closed degradative system and restoring the sieve before the switch is thrown.